I'd like to welcome you all to NAISMA's August webinar. Today's webinar is Research Spotlight Managing the European Green Crab Invasion. And before we begin, I'd like to share a little bit about NAISMA for those of you that don't know us well. So NAISMA is the North American Invasive Species Management Association. We are a nonprofit organization, and our mission is to empower invasive species management across North America. And we are here to serve members in the professional and academic world who work on invasive species. We have 18 board members and 10 active committees who support our programming. We have 7 team members and a membership that continues to grow every day. We communicate with over 15,000 subscribers and interact with over 38,000 social media followers. And during National Invasive Species Awareness Week, we were able to extend that reach to over 3.4 million people, which is very exciting. So what do we do? Well, NAISMA provides the connection, the tools, and the voice you need to address invasive species. We frame our programs around these core pillars. We work hard to provide opportunities for collaboration and connection through our webinars, summits, and annual conference. We provide a collective voice on a national platform to advocate for relevance and support through our government relations program and National Invasive Species Awareness Week. And we equip our membership with the tools needed to manage and educate the public about invasive species through Play Clean Go, the NAISMA Shop, our certified weed-free products program, mapping data standards, and NAISMA Academy. An event you won't want to miss is the 2025 NAISMA Annual Conference. So please mark your calendar and plan to join us in Lake Tahoe from November 3rd through the 6th. Our theme this year is charting a new course. And we have some great speakers lined up already and hope to see you there. You can register at naisma.org. We also hope to see you at our Invasive Upcycling Summit that will be taking place next Wednesday, August 27th from 11 to 3 p.m. Central. This event, co-hosted with Invasive Impact Initiatives, will bring together changemakers who are transforming invasive species into useful, sustainable products by collaborating with local invasive experts. They're also educating communities about the ecological damage invasive species cause and inspiring new approaches to environmental care. And you can view the agenda and register for this event at naisma.org. All right. As a friendly reminder, your membership dues help to fund our educational programs like this webinar. If you are not currently a member, we offer a variety of different membership options. You can view our new membership structure on our website. And then if you want to sponsor NAISMA in a bigger way, our new Premier Partner program is designed for you. Premier Partners enjoy benefits throughout the year, including acknowledgement on our website, at events, and more. And so we would like to acknowledge and thank all of our 2025 Premier Partners, supporting agencies, and foundations whose generosity helps to make events like this webinar possible. So for everyone watching, please be sure to put any comments or questions in the Q&A box and not in the chat box, so we can be sure to answer everyone's question after the presentation. Do take a look first and see if your question has already been asked, and if so, give it a thumbs up to promote it to the top of the list. And so to get us started with the main event, I'd like to introduce you to Tim Campbell with Wisconsin Sea Grant, who has helped us to organize this event. Tim is the Aquatic Invasive Species Program Manager for the University of Wisconsin Sea Grant Program and the National AIS Liaison for the NOAA Sea Grant Program. He supports AIS prevention, outreach, communications, and program evaluation, and works to apply university and sea grant resources to AIS issues. And he's been with Wisconsin Sea Grant since 2011. So Tim, thank you for joining us today.
Great. Thank you, Tegan, and thank you, NAISMA, for helping us organize and host this webinar on invasive green crabs, and thank you everyone for joining us today. This is our second in a series of 2 webinars on green crabs. Sea Grant is doing work across the country on green crabs, everywhere from harvest and markets for green crabs on the East Coast, kind of early detection along the Pacific Northwest, and responding to new invasions in Alaska. Sea Grant's working with communities across the country on green crabs. And on the second webinar, we really were interested in trying to pull together some current research on European green crabs to help us just understand some of the latest science happening in this space. And so we'll start kind of big picture on a transition from resistance to acceptance in managing invasions, and try to get more specific as our webinar goes on. So If it works for you, Tegan, I'll introduce speakers as they're about to share their slides, give us a little bit of time between everyone. So at that, I will bring up Abby Keller. Abby is our first speaker, and she's a PhD candidate in environmental science, policy, and management at the University of California, Berkeley. And she studies decision analysis and quantitative ecology. And today she will be our first presenter in presenting on the transition from resistance to acceptance, managing a marine invasive species in a changing world. And Abby, feel free to share your slides.
Yeah, like Tim mentioned, I'm Abby Keller. I'm a PhD candidate in California. I've been studying green crabs since 2019. I did my master's at the University of Washington, so kind of it's such a rich subject matter. So it feels like there's so much interesting research to be done. I'm going to share some of my work applying concepts from decision theory to help invasive species managers make better decisions when there's a lot of uncertainty. I just want to first highlight my co-authors, Tim Cunahan from USGS, Ted Grossholtz from UC Davis, and then Carl Boediger, who's my advisor at Berkeley. As you probably know, this talk is in the context of the European green crab, which is commonly cited as one of the world's most impactful invasive species. They commonly have two main modes of impact. They're quite aggressive predators. I think Mary's going to be talking about what they're eating more specifically, but they tend to consume shellfish that are often commercially valuable. Also, while they're foraging, they can uproot eelgrass beds, which are really valuable habitat for juvenile fish and shorebirds, and also play an important role in shoreline stability. So it's kind of quite a high priority species in many coastal areas. So the green crab is found on all continents except Antarctica, but my research focuses mostly on the Northeast Pacific, so kind of mostly Washington State. For most of the last 20 years, the green crab has been present, but at quite low abundance, kind of following this boom and bust cycle. So this chart here, you see the last 20 years, mostly with kind of the catch rate, see that's quite low. But then in 2019, there was a bit of a regime shift where crab abundance really skyrocketed in multiple bays around Washington. These coastal Washington bays produce 25% of the country's oysters. So for shellfish growers in the region, this change was a huge threat to their livelihood and to their culture. Multiple tribes also had pretty huge outbreaks on their tidelands, including the Lummi Nation, who declared this an environmental disaster. So to kind of respond to this existential threat, the governor of Washington declared a state of emergency. But the pace of change in these new circumstances were introducing a lot of new uncertainties that were kind of straining the ability to make rational decisions. And it turns out that managing the species can be really difficult. So the region is fragmented by tribal, state, and federal jurisdictions. There's also quite a bit of private land. So you have individual shellfish growers who are all making their own decisions about controlling green crab. But the problem is we have kind of this contrast between the really small spatial scale of decisions and a really large spatial scale of the ecological process. So while adult green crabs, they live kind of in bays and estuaries, they have a planktonic larval stage where they move out into the open ocean for about 60 days. And during certain oceanographic conditions, they can travel hundreds of kilometers away from their source. So you can imagine if you're a manager of a bay, you can be removing the adult green crab, but they can really easily recolonize from neighboring areas due to their long distance dispersal and kind of this influx of non-local propagules. So to kind of build a bit of intuition further about how green crab populations respond to removal, so you can imagine a scenario where you have a quite isolated bay. There's mostly local reproduction, local propagules. We can think of two contrasting scenarios, one where you put in a low level of removal effort, one where you put in a high level of removal effort. The remaining adults will produce local propagules. Inevitably, there's a few coming from somewhere else. These kind of larvae or juveniles undergo density-dependent recruitment into the population. And we see that the resulting density is closely coupled to the level of removal effort. So kind of a different scenario is more kind of a connected population where you have both local and non-local propagules. You can again put in a low or high level of removal effort. The remaining adults will produce local propagules. You have the same number of non-local propagules entering both populations. And then after recruitment into the population, we see that the resulting density is now decoupled from the level of removal effort. So this research was part of a larger structured decision-making effort, where the first step of this process is to characterize the decision problem and frame the context. So I interviewed kind of many decision makers throughout the region to get a sense of the types of decisions they are making and the challenges they were facing. And for many decision makers, controlling green crab feels like this Sisyphean task. They're wondering if their removal efforts are futile and if they're just going to be sinking money into this problem forever. And these questions and this kind of way of thinking are increasingly common in 21st century ecosystem management, where managers are often facing really rapid ecosystem transformations that can really benefit from thinking beyond just the traditional focus of resisting change. So this paper by Abby Lynch in 2021 introduces this concept of a RAD approach that's kind of meant to support decisions that are strategic in addressing kind of profound change and rapid transformations. And we found that this framework is really applicable in this context. So here we're framing this green crab decision problem as this choice between resisting change by removing the crabs and trying to, with the goal of conserving either current or historical conditions. Or the alternate strategy is accepting that this is a trajectory that is beyond our control. We can't change it. We should instead invest these resources in adapting to a new normal. But for green crab, this question of whether you should be resisting or not resisting, it's hard to know. So there's a lot of uncertainty around rates of population growth, colonization, and even the effectiveness of removal is really not well characterized. So here comes in decision theory, which is a really useful way of thinking when trying to make rational decisions under uncertainty. And essentially, decision theory both helps us understand how to break down and describe the problem, and also provides a set of methods and formulas that we can use to optimize decisions under uncertainty. And one of these kind of methods or way to formulate the problem as a model is a Markov decision process that's useful for sequential decisions. So you can imagine every year you're making a decision about how hard you should try to remove green crabs, and you make this decision every single year. So that would be a sequential decision. And this type of model takes an action. So here, this would be the annual removal effort that affects the system state, which here is the green crab density of the local population that you're managing. So the way that this kind of model works is, let's say today the system is in state A. You take an action. This action causes the state to transition to a new set of states with different probabilities associated with those transitions. And again, this process continues. You again take an action next year, which again causes the kind of states to transition to a new set of states, kind of so on forward in time. Imagine doing this, iterating this for hundreds of years. So what we're looking for is the action to take today, considering all of the trickling ways that today's decision could influence the future state. So to make this green crab relevant, we first built a stylized model of population growth, colonization, and removal. When I say the word stylized, to mean that it's relatively simple. So we're kind of focused on capturing the main processes, drawing parameter values from the literature. But, we're also kind of accounting for stochasticity or randomness in these rates. So for example, the colonization rate can vary dramatically from year to year based on factors like ocean temperatures or conditions like El Nino events. Some years you have lots and lots of larvae entering your bay, other years you have very few. So we're accounting for randomness in the process. We're also accounting for uncertainty in the parameter values themselves. So for example, this plot here on the right shows the relationship between the removal effort and removal rate. And we know that the maximum removal rate is likely less than 1, since these crabs are caught by traps that are size selective. But the exact maximum removal rate is uncertain. So we're accounting for this parameter uncertainty in the way that we've formulated the model. So we use this model to quantify how a population responds to removal and relate the local equilibrium population size to the level of green crab removal effort. So here on the x-axis, we have the equilibrium size of the local population. So of course, this population size varies from year to year based on variable growth rates, mortality rates, variable colonization rates. But we can think of this equilibrium as kind of the long-term average. And we're also accounting for uncertainty in these rates. So there's some probability associated with the equilibrium size. So let's first consider a regime where the colonization rate is low. So this is kind of an isolated population where there are very few non-local propagules. If you do nothing or apply a removal effort of 0, the population will reach carrying capacity, which you see signified as K on the x-axis here. And as we increase the level of removal effort, we see that the equilibrium population size is coupled to the level of removal effort. So high removal effort equals lower population size. We can consider a different regime where the colonization rate is much higher. So this is a more connected population where there are many non-local propagules. We see that as we increase the level of removal effort, we really can't knock back the population size. So this kind of is a scenario where the population size is decoupled from the level of removal effort. So this kind of is a quantitative representation of kind of the feeling of futility that a lot of managers in the region feel. So the next question is then, under what scenarios do we resist, and then when do we accept? So we're embedding this model within an optimization framework. We're thinking about this as a multi-objective question. So here we want to be minimizing multiple objectives. And we're finding the removal effort or the action that minimizes both kind of the cost of removal and minimizes ecological impact. And here the cost of removal is a function of the action, the removal effort, where lower removal efforts have a higher value. So essentially it's more costly to have a higher removal effort. And whether this relationship is linear or nonlinear kind of depends on the constraints of the decision maker. And the amount of ecological change is a function of green crab density, with lower green crab densities having higher value. And this relationship is likely nonlinear and probably depends upon the impacted species. So maybe the shape is different for shellfish than it is for eelgrass. Because this is kind of a multi-objective problem, we have to weight these two objectives. So this is a way to formulate the problem that allows a decision maker to express their values. So here this reflects a scenario where a decision maker cares much, much more about minimizing ecological change than minimizing the cost of removal. And before I get to the results of the optimization, I just want to clarify a symbol that you'll see. So what we're describing as this decision boundary between resistance and acceptance, it's really sensitive to the colonization rate or kind of the size and level of connectivity to source populations. So this is signified with the symbol alpha. So an alpha of 0.5 indicates that the source population is half the size of the carrying capacity of the local population. And an alpha of 1K, would indicate that the source populations are equal to the size of the carrying capacity of the local population. So we're accounting for the fact that the size of the source populations and level of connectivity is variable and varies from year to year. So this value is more of a summary statistic or kind of an average kind of abundance and connectivity over time. So should I get ready for the results? Here, we're showing the optimal level of removal effort on the y-axis as a function of the local green crab population size on the x-axis. And these three panels moving from left to right correspond to increasing kind of size and level of connectivity between the source population and kind of the local managed population. So when alpha is small, so this is an isolated population where kind of connectivity to source populations is low, it won't surprise you that the optimal strategy is to continue resisting. So here, this means that the optimal removal effort is greater than 0. But as alpha increases, we see this kind of switch between resistance and acceptance, where for kind of larger local population sizes closer to carrying capacity in the dark gray, the optimal removal effort is 0. So that kind of means the best strategy is to accept. Whereas at lower local green crab densities in the lighter gray, the optimal removal effort is greater than 0. That means it's optimal to continue resisting. And then as alpha continues to increase, we kind of see this shift where it's optimal to accept for all local green crab densities. Kind of essentially, this is the scenario where this is a trajectory we can't control, and resources should better be invested in adaptation. So I just kind of want to revisit the way that we've set up this problem, because the question of whether to resist or accept is really a reflection of the values of a decision maker. And they express these values in how they weight these two optimization objectives. So again, previously we considered a scenario where a decision maker cares much, much more about minimizing ecological change than minimizing the cost of removal. But what if we change these objective weights, where this reflects the values of a decision maker who cares equally about these two objectives? So for comparison, I've put the previous results on the top here. So again, these panels moving from left to right correspond to increasing levels of connectivity. And this bottom panel shows kind of results for these new objective weights, where a decision maker cares equally about these two objectives. So in this scenario where kind of you have a low alpha, you have an isolated population, it is still optimal to continue resisting. So this is an optimal removal effort greater than 0. But now we see as alpha increases, so an alpha of 0.5K means that on average, your local population is connected to a population that's half the size of the local population. But in here, it becomes optimal to accept for all kind of local green crab densities. And then increasing alpha further, obviously everything greater than this, it's kind of still optimal to accept when you have the larger source populations. So we can see that the answers that you get in these optimizations really depend on the way that the objective function is set up. So we built this kind of interactive web tool, our Shiny app, where a decision maker can specify their own objective function and their own weights and run these same optimizations. So this model accounts for different types of uncertainty and kind of includes randomness or stochasticity in the system, but it also assumes that you know the size of the source population. For example, in this figure on the left, we're allowing the source population or like intensity of colonization to vary from year to year. So some years it's high, some years it's low. But we're assuming that you know the shape of this curve, you know the mean and the variance of the size of the source populations, which is definitely not something that's totally obvious in the real world. But I just want to highlight some recent oceanographic larval modeling work by Lily Engel, where she's characterized levels of connectivity among populations in the region. So the results of our kind of decision analysis exercise provides a useful context for understanding how to incorporate this new information into decision making. Which gets me to some concluding thoughts on how I see a model like this informing actual decisions. So, like all models, the one we've developed is definitely a simplified version of reality, and we really don't anticipate decision makers using this to make really like detailed, granular decisions. But we think this is really useful as a way for decision makers to build intuition around the problem. Anecdotally, we've seen that these results have been used to build consensus about how the problem of green crab management should be framed as this kind of contrast between resistance and acceptance. We found that it's quite uncomfortable to use the word accept. It's, like a bit taboo to acknowledge that the pace of change is just so fast and beyond our control. But often, managers are implicitly making the choice whether it's worth it to continue to control invasive species. But here we're making this question explicit and applying formal decision analysis methods to help decompose this question and show that under quite realistic scenarios, it's probably likely better to adapt than to resist. Yeah, that's basically everything. I'm totally happy if you want to chat about any of this further. If you have any questions or concerns, here's my email. If you're interested in the Shiny app, there's the link, and then below is the DOI for the paper associated with this talk.
Thank you so much, Abby. Just a reminder for everyone, we have the Q&A box within Zoom, so please put your questions there. The speakers and the panelists will be happy to answer questions as we can through that during the presentations, and then we'll have time after all the presentations for a more general question and answer session. So thanks again, Abby. We'll move on to our next presentation with Mary Fisher. Mary is a postdoctoral scholar at the Coastal and Marine Sciences Institute at the University of California, Davis. She is an interdisciplinary marine scientist interested in how adaptation to climate change affects coupled human-natural systems. Her research is drawn on quantitative and qualitative techniques to explore trade-offs and unexpected consequences associated with climate adaptation in U.S. West Coast fisheries. Today, she will be presenting What Can Diet DNA Tell Us About Predation Impacts of an Invasive Marine Species? Mary, the floor is yours.
Thanks, Tim, for the introduction. So I'm currently a postdoc at the University of California, Davis, but I'm presenting research today that I did with Washington Sea Grant's crab team while I was a graduate student at the University of Washington. And we were funded for this work by the Washington Department of Fish and Wildlife as part of the Willapa Bay Oyster Reserves Grant Program, and we're also supported by the Willapa Grace Harbor Oyster Growers Association. And Washington Department of Fish and Wildlife and the Oyster Growers Association wanted to understand the possible predation impacts of European green crab. The reason that we're all here today. I want to thank Abby Keller for giving a great introduction to green crab and their impacts on coastal ecosystems so that I can kind of dive right in. I'm going to go into detail about green crab in Willapa Bay, Washington, and the findings of our research, but I'm also going to try to speak more broadly about when and how to use DNA-based diet analysis methods in general. So I want to start by setting the stage for our study in Willapa Bay. Willapa Bay is a large estuary on the coast of Washington state. It contributes to approximately 1/4 of all Washington state shellfish aquaculture production, primarily from Manila clam and Pacific oyster harvest. Green crabs have been present in Willapa Bay since about 1998, but in 2015 through 2017, local shellfish growers and scientists observed a really dramatic increase in both the relative abundance of green crabs as well as their geographic range within the bay. And this, of course, raised concerns that green crab might already be reducing the harvest of some of those really important manila clams, and that they might be having other impacts to native and commercially important species. So we weren't just interested in what green crab were eating in Willapa Bay, we also wanted to know whether they were eating differently when they were on cultured manila clam beds versus uncultured slough sites. And to determine this, we worked with clam growers and partners in the green crab trapping and removal program to sample European green crab on 2 active clam beds in Willapa Bay, Oysterville and Long Beach, and those are going to be the green points on the map. And then we also sampled green crab from 2 Washington Sea Grant crab team monitoring sites at natural sloughs in Stackpole and Nakata. And those are going to be those orange points on the map on the slide. So all 4 of these sites had about 20 traps set per tide and collected over 300 green crab in total. We obviously had to select a smaller subset of these 300 to conduct our analysis. And in order to select that subset, we hypothesized that larger male green crab would have greater crushing strength in their claws and were therefore potentially more likely to be able to use manila clam as a food item. And I'm just showing on these graphs here on the right the size distribution of all trapped green crab in gray versus the green crab that we use for our analysis in red. Ignore the black bars on these graphs for now. The x-axis is carapace width in millimeters, and the size distributions are broken down by the 4 sites that we trapped crabs at. And hopefully you can see here that the red bars on the graph tend to be on the larger end of the distribution of sizes. The mean carapace widths of the crab that we analyzed were about 66 to 67 millimeters. And then we further hypothesized that because higher temperatures tend to increase metabolism, there might be more feeding activity. Crab might be more active on clam beds in the summertime, making them potentially easier to trap and maybe more full. And so we focused on larger male green crab eating on and off clam beds May through September, so during some of the warmer summer months. All right, so we have all these decisions that we made in our study design about what green crab we're going to look at. How do we actually test them? The traditional way to directly observe diet is visual observation, right? So you open up the stomach or cause the organism to regurgitate its prey and look at what's there. This is a pretty quick and inexpensive method that works really well when stomach contents look like these photos of groundfish stomachs that were taken by the Northwest Fisheries Science Center survey team. But this is what the stomach contents of a green crab look like after you've dried them. We also get a lot of liquid digesta that looks a little bit like a smoothie for those of you who just finished eating lunch. And this is because crab not only shred their prey while they eat it, they also have a special instrument in their stomach that continues to crush and grind their prey after ingestion. So as you might imagine, visual identification of what's in these stomach contents is usually lower resolution. We might be able to identify classes of organisms unless you're really an expert with a lot of experience in the local ecosystem. You also run the risk of missing prey items that are composed of soft tissue or prey that have already been highly digested. And this might be of particular concern if the way that you're sampling is by trapping predators instead of hand collecting them, because that digestive process is going to continue while the crab is sitting in the trap. And in these cases, you could turn to diet DNA, which is more technically called DNA metabarcoding of the stomach contents. The pros of diet DNA is that you can get higher resolution for certain species like crab. You can identify soft and highly digested prey. And you can sometimes design these methods to be a little bit more sensitive to prey groups that are of interest to you. You do have to account for the fact that diet DNA does tend to be a little bit slower and more expensive than visual observation. Okay, so if you're looking at this direct column and thinking like, what about stable isotopes or fatty acids? These are indirect methods of describing diet that can also resolve to lower taxonomic levels if you have isotope or fatty acid signatures of other species in the ecosystem. These are a whole separate set of methods that I'm not going to get into, except to just say that combining direct and indirect methods of describing the diet can provide a really particularly powerful insight into predation by invasive species. Before I dive into the technical talk of DNA metabarcoding and diet DNA, I am hoping that you all can answer a very quick poll just to give me an idea of how familiar everyone on the call is with diet DNA, DNA metabarcoding, or eDNA. Awesome. I'm already seeing a lot of thumbs up and hearts traveling through the air. So if you go to your reaction button, a heart is not at all familiar. Thumbs up is fairly familiar, and then the little party icon is very familiar. I see a few of those as well. Okay, great. It looks like we have a pretty high diversity of knowledge in the room, but most people at the not at all are fairly familiar. So I'm going to walk through our process of doing diet DNA analysis on a green crab stomach. So the step one is obviously green crab consumed their prey. In that process, they shredded that prey into tiny pieces. Sometime after they ate, they were trapped and transferred to the University of Washington, where I dissected them to remove their stomach contents. I then extracted DNA from their stomach contents, and the DNA from stomachs are pretty degraded because it's been broken down by digestive enzymes. And as a result, we get small amounts of usable DNA. And the part of the DNA that we're actually looking for is a pretty small fragment called a barcode. For this study, we were interested in a fragment of DNA within the CO1 region of the mitochondrial genome. That's for those of you who threw up those party icons before and might be curious in the finer details of what we did. For the rest of you, this is just generally the more commonly used barcode region for animals. Step two is, in order to have enough DNA to actually do our analysis, we need to make a lot of copies of that barcode. And we do this using a copy and paste process known as polymerase chain reaction, or PCR. And that's the same PCR that's used in COVID testing. So the PCR that we use gives us enough DNA to actually work with, but it can also be a little bit selective. And so I like to kind of compare it to a fishing net or a trap. You're targeting a subset of the animals that are actually present in the ecosystem. And as a result, it is helpful to do a little bit of testing before you start diving into your samples to kind of determine what subset of organisms you'll end up catching in this net, especially if you have organisms in mind that you want to look for, like we did with manila clam. So for our study, we ended up choosing a net or a PCR primer that was designed to capture benthic invertebrates best, although you'll also see that it captured a diversity of other organisms as well. And we knew that our PCR primer or our net would capture some of our important prey species just because we did extra lab work at the beginning of this research to test the affinity for manila clam, Pacific oyster, and some other economically and ecologically important species. And then in order to be confident in our results when we're working with our samples, we also complete this copy and paste process 3 separate times for the same crab. So if you have 100 crab, you're getting 300 lab samples with 3 replicates for each crab stomach. All right, step 3 is that we send our DNA through a machine to be sequenced. The machine outputs DNA sequences as long strings of the letters A, G, C, and T, repeating in different patterns. And in step 4, we match those sequences to a database, which tells us which organism that DNA actually came from. Okay, before I kind of go from methods to results, I want to premise our results by doing a quick recap of what diet analysis can and cannot tell us. And this is diet analysis in general. And I'm going to use manila clam in these examples because that was one of the species that we were particularly interested in. So diet analysis can tell us things like green crab are eating manila clam. Manila clam is more prevalent in the diet at Long Beach than Stackpole. So comparing prevalence in crab stomachs between sites. And then we can also look at the prevalence between different species. So we could say manila clam is more prevalent in the diet than softshell clam. And again, these are just examples here. Diet analysis can't rule out that green crab never eat manila clam. We're always going to be selecting a subset of individuals out in the ecosystem, so we can't rule out that they're eating something. We also can't differentiate when green crab ate that prey item, so we couldn't say that green crab ate manila clam inside or outside of our trap. However, we can take data on what organisms were in the trap and if there were evidence of predation on those organisms while we're collecting green crabs, which we did. And then we can't actually count the number of individual manila clam that a green crab consumed, right? And we're just going to add one more thing to this category on the right-hand side here that's specific and important for diet DNA, which is that we wouldn't be able to say that green crab ate more of manila clam than softshell clam. So we can't compare the total quantity of a prey item that was eaten by an individual green crab. And I've just put an asterisk there because if I have time at the end, we're going to go into some of the ways of getting around this limitation. But for now, for the results, I just want to set the stage and premise the types of questions that we can and cannot answer with this analysis. All right, enough with the methods. What are green crab eating in Willapa Bay? We identified 54 unique prey taxa in 61 green crab stomachs, and we were able to resolve 90% of these down to the species level. So just as a comparison, some of the previous visual diet studies of green crab have examined between 600 to 800 crab stomachs, so an enormous amount of work, and resolved 20 to 25 food items. So the number of prey taxa we observed is speaking both to our methods and, of course, to the eating habits of green crab here. The most frequently observed prey group that we found in the stomachs was arthropods, although we really observed a wide diversity of different prey items from worms to red algae. And I've listed all of those prey groups on the horizontal axis of this graph. And to explore the frequency of occurrence of all prey items, we calculated the percent of crabs that had that prey item in its stomach, and that's on the y-axis here. So here, what I'd like you to see is that individual prey items generally had lower frequencies of occurrence below 10%, and that's just because of the huge diversity of things that we found in green crab stomachs in Willapa Bay. So for example, there were a number of different mollusks present. That's going to be number 6 on the horizontal axis, but the frequency of each individual taxon across crabs ranged from pretty rare to a little bit more common. And this is very much emphasizing green crabs as generalist predators. But we did, as you might have noticed here, have a few prey items that had relatively high frequencies of occurrence. So for example, 21% of the crab that we looked at had consumed the native hairy shore crab, Hemicrapsis oregonensis. And we know from California research that native hairy shore crab numbers have declined as green crab become more abundant. So that kind of aligns with previous research. 14% ate red algae, specifically in the genus Neoporphyra, and 12% had consumed the Pacific staghorn sculpin, which is also a native and ecologically important local species. Also within our kind of top 10 most prevalent species was a sand shrimp and a non-native amphipod. You'll notice that the manila clam did not show up on those most prevalent lists that I went over, and that's because we only observed one green crab stomach with manila clam in it. This individual was trapped at a clam bed. It's at our Oysterville site in September, and I've shown that on this graph right here by listing our site names on the horizontal axis and then our species on the vertical axis, and that green point is going to indicate that Oysterville is a clam bed, and that's where we saw our manila clam. We also kind of looked into some other locally important species or species that have been impacted by green crab in other places. That's going to include the soft-shell clam, Maya arenaria. So we saw evidence of fairly high, relatively high percentage of green crab at all of our sites consuming soft-shell clam. I also want to point out that 6 crab across the 4 different sites had DNA from the families Asteraceae in their stomachs, and that's going to include eelgrasses and surfgrass species. So both of these are found in Willapa Bay, and we would have to do more research to determine which species those are, especially if it's the native eelgrass species, and whether crab here are consuming eelgrass intentionally or maybe accidentally while foraging for prey. And then the other species I'd like to point out here is Dungeness crab, which is an important commercial and recreational fishery species. And we observed Dungeness crab in the stomach contents of green crab at 2 different clam bed sites. One of the things I hope you start to see from this graph is that we did observe some differences in stomach contents across the clam bed and the uncultured slough sites. And so we ended up conducting a principal coordinate analysis in order to more statistically determine the difference between our clam bed and our slough sites. And then this 2D space here, the points that are closer together represent crab with more similar stomach contents, and those farther away represent crab with more different stomach contents. And those clam bed sites are going to be shades of green, with the slough sites shades of brown. And hopefully what you're seeing on this graph is that the 2 clam bed sites and the 2 slough sites kind of cluster more closely with each other in the coordinate space than sites of the opposite type. And we conducted some statistical testing. We did find that there were significant differences in stomach contents between green crab trapped at one of the slough sites and those trapped at the clam bed sites. And there's no statistically significant difference in stomach contents between sites that were both clam bed or both slough sites. Okay, so as a recap, our data shows, I think, first and foremost, how green crabs are generalists. They consumed a wide range of native and non-native prey items across a huge number of phyla that were present in the ecosystem. Particularly, several native species stuck out in the stomach contents, and that includes the hairy shore crab, the Pacific staghorn sculpin, and a sand shrimp species. And these are candidates for experiencing population level impacts that ongoing research by Washington Sea Grant crab team is exploring further. And then, of course, speaking to one of the main motivations for this study, we only observed 1 out of 86 crabs eating manila clam. And I just, on that particular point, I want to recall that we can't say what green crab are not eating. So next steps might include looking at other size classes, looking at female crab instead of predominantly male crab, and then also looking at stomach contents from other times of the year. And I think that I am at time, so I'm going to skip through this extra material and just say thank you all for listening. Thank you to the Oyster Reserves Board for selecting our project for funding, to the Willapa Grays Harbor Oyster Growers Association for working with us to identify clam beds and helping to actually trap these crabs out in the field. This QR code here is a link to our open access article that reports on all of these and more results from the study. And you can always reach out to me via email. That's a stable email address that won't go away because I'll be leaving UC Davis in the next couple of months. All right, thanks everyone.
Thank you so much, Mary. Hopefully somebody asks you a question so you get to use all those extra slides you have. It'll be fun to see what's there. As a reminder, we have the Q&A option open on Zoom, so please ask your questions there so we can either answer them during the presentations or afterwards. And so lastly, we have Rikke Jeppesen up to present. She is currently an environmental scientist at the Central Coast Regional Water Quality Control Board in San Luis Obispo. Rikke has a PhD in ecology and evolutionary biology from the University of California, Santa Cruz. Her green crab studies were conducted at Elkhorn Slough National Estuarine Research Reserve, where she studies invasive invertebrates, while also working on water quality research and large-scale salt marsh restoration projects. Her presentation today is titled Recovering Population of the Southern Sea Otter Suppresses the Green Crab, a Global Marine Invader. Rikke.
Thank you so much for the introduction. And thanks everyone in the audience for your time and attention. I'd like to start by pointing out this was very much a collaborative project. And today I am just the spokesperson for a group of amazing people. As Tim said, this work was conducted at Elkhorn Slough Reserve, and I'm now with the Central Coast Regional Water Quality Control Board. So we used long-term data to shed light on a puzzling pattern. And I'm going to start by introducing you to the first main character of today, the southern sea otter, which is found in various places, but one of them is Elkhorn Slough Reserve. It was hunted for its fur, and it was at the brink of extinction in the early 20th century. However, today it is a recovering apex predator. An apex predator is the key player in trophic cascades. The trophic cascade is this phenomenon that you have a complex food web. And if you remove or add more of the top players, this top predator has the ability to exert top-down effects on the organisms below it. So the effects can be direct with a solid arrow and negative. For example, an otter eats a crab. It is direct negative effect on the crab. Effects can also be negative and indirect. For example, as everyone eats each other, at least from top to bottom, if the otter eats crabs and crabs eat these sea hares, then by removing the crab, you have more sea hares. It means sea hares eat more algal epiphytes. So in this case, the otter has an indirect negative effect on epiphytes. The otters can also have indirect positive effects on other organisms. So while we could talk at this at length for the whole day, I'm going to focus on these two top players. And the second one is in this diagram. It's a crab, and it's not just any crab we're talking about. It is the European green crab for today's talk, which is a globally invasive species. So invasive species populations exhibit certain characteristics. One is that of rapid population growth. So over a relatively short amount of time, the population goes from low to maybe exponential growth, and population growth is high. Invasive species also exhibit rapid range expansion. So you find it in one place and all of a sudden it's all over the place. And just like the otter can affect species below it, the green crab or invasive species in general can alter the existing communities that they enter. So if you have a community of species before introduction of an invasive species, you introduce the invader, and the relative abundances of various species may or may not change after that. So the specific invader that we're going to discuss today is the green crab, and I'll tell you a little bit about its history. It is native to Europe, found from Norway down to northern Africa. It has been on the US East Coast for a couple hundred years, and on the West Coast, it's been around for 30-ish years. In 1989, it was first detected in San Francisco Bay, and by 1994, Ted Grosholz discovered it at Elkhorn Slough Reserve. Shifting gears to Elkhorn Slough. Elkhorn Slough is a tidal estuary found at the central coast of California in Monterey Bay. At Elkhorn Slough, we find both otters and green crabs, and we have long-term monitoring data for both of those species. As for the otters, Elkhorn Slough Safari, which is a tour boat company, have been making their visitors count otters every day for decades. Also, USGS has annual counts of otters in this area. And USGS also has foraging observations of otters, meaning they look at what otters eat through really high-powered scope. As for crab monitoring data, Elkhorn Slough Reserve has had long-term crab monitoring since early 2000s. We visited 9-ish sites. Over 20 plus years of data have emerged. And when we look at the data from the otters and the crabs, we find that, so time is on the x-axis, relative abundance is on the y. When otters are abundant, which they have been in recent years, then green crabs are not abundant. The blue line is relative green crab abundance. When otters peak, green crabs are at their low and vice versa. So we were wondering, does that mean could the otters be controlling this green crab population? So we used long-term monitoring data, and we looked at the spatial and temporal relationships between these two species at a local and a coastal scale. So we know that otters eat crabs in general, because from scat, we've seen crab parts in otter scat. But do they eat green crabs? And if so, do they eat enough to make a dent in the population of the green crab? So the first research question was, do otters consume green crabs at Elkhorn Slough? The answer is yes. We have 3 lines of evidence. Not only does the culprit get caught in the act by photographers, as I mentioned, the USGS observers look at otters with scopes, and they can actually identify some prey down to the species. And last but not least, one of my co-authors was out working at the slough one day. In the background, he hears this loud chewing noise, and he turns around to look at what it is. It is an otter just mowing through one green crab after the other, and it ate about 30 in 1 hour. So yes, otters eat green crabs, but how many do they eat? So another of my co-authors, Tim Tinker, did a back of the envelope calculation to figure out how many green crabs can the otter population in Elkhorn Slough as a whole consume. And he did all these calculations. The point I want you to focus on is that about 300-ish green crabs could disappear every day from the otters. It still doesn't answer the question whether that's enough to make an impact on the green crab population. So Tim did some modeling. He developed a Bayesian model and looked at the spatio-temporal dynamics of sea otter and green crab populations at the slough. The results are not easily digestible, so I'm going to walk you through just one site. So we have good data from otters and crabs from these 6 sites in the 6 panels for about 20 years. The dotted line is the otter density, and the solid lines are green crab densities. Red is in the absence of otters in the model. Green is in the presence of otters. So the point here is that if the red line is above the green line, it means that increasing otter density can lead to a decrease in green crab abundance, which you can see is the case for the 6 panels. The red line is in general higher than the green, above the green line. So the next question we asked ourselves was whether otter and green crab abundances vary with tidal restriction. Why do we care about tidal restriction? Well, Elkhorn Slough is a very impacted estuary from human activity. More than 50% of the historic estuary, all the gray areas, are actually behind water control structures such as tide gates, culverts, the railroad, roads, you name it. So the only areas which are the ones in blue that still have full tidal flow are the harbor and the main channel. And from past monitoring data and from green crab studies in other places, we know that sites with restricted tidal flow can have poor water quality, and those sites are particularly well suited for green crabs, it seems to be the case. So we wanted to investigate that pattern at Elkhorn Slough. So we looked at sites that are tidally restricted in red or blue, non-restricted at Elkhorn Slough, because that is the most prominent habitat difference there. And what we found was, sure enough, when you have full flow, no tidal restriction, you find a lot of otters, not nearly as many green crabs. Look at those tidally restricted sites, otter density is very low, and green crabs are, I don't know, not through the roof, but sometimes they are. So moving on from this local scale of Elkhorn Slough, we wanted to zoom out and think about how that Elkhorn population is doing relative to other populations up and down the coast, because, or mostly up, because we have neighboring bays and estuaries. And the question we tried to address was whether Elkhorn Slough is different from estuaries without otters, because, as I said, at Elkhorn Slough, we have green crabs, we have otters. These other bays and estuaries, there are green crabs, however, there are not otters. So what we found when we compared our green crab numbers to our neighbors were, and I apologize, this graph is a little messy. We still have time on the x-axis, and then we have relative green crab abundances on the y-axis. The red line is Elkhorn Slough. So overall, you can see green crab populations fluctuate. However, at Elkhorn Slough, the red line, it has decreased and remains low in the past, I don't know, 7-ish years. Whereas the other estuaries and bays that do not have otters, the green crab populations also fluctuate like everywhere, but they're generally higher than they are at Elkhorn Slough. So while green crab populations seem, I guess, higher in the areas without otters, they're definitely lower in the areas with otters. So just to sum up what I went over is, do otters consume green crabs at Elkhorn Slough? The answer is yes, and it's enough to affect the population. Secondly, how about the tidal restriction? Yeah, you find more otters and fewer crabs when you don't have tidal restriction, which is what you'd want. Tidal restriction is not very beneficial for the system. And last but not least, Elkhorn Slough is different from estuaries without otters because the green crab population at the slough remains lower than other places in the presence of otters. So why was it important to answer these questions for us? We can use this information to manage for multiple goals. We can support apex predators to maybe better control invasive species, such as the European green crab. You heard from the first talk today that invasive species are really difficult to manage for. Or restoring sites with tidal restriction and restoring them back to their former hydrology by removing those tidal restrictions could actually decrease that ideal habitat for invasive green crabs. And restoring lost marshes or eelgrass beds can benefit the food webs that then benefit the otters and increases just resilience of the system as a whole. So what we learned from this work was that understanding species interactions can really contribute to ecosystem conservation. Supporting threatened apex predators, the otter, while limiting invaders, that is just a win-win scenario. And enhancing this ecosystem function can provide resilience against future threats to our valuable systems. So protecting these important ecosystems that are already strained by human activity will make the natural habitats with their associated flora and fauna available for future generations to enjoy. This work, of course, could not have been done without tons of help from staff, from researchers, from volunteers, multiple sources of funding, and we are all eternally grateful for everyone who pitched in to make this happen. So hopefully I have conveyed to you that sea otters can keep invasive species, such as the European green crab, in check. It is of utmost importance that we continue to restore and protect essential marsh and seagrass habitats for the benefit of this incredibly cute species. So with that, I will hand it back to Tim. I'm not sure how questions will be managed. Thank you all.
Thank you, Rikke, and hooray for the otters, and thank you for sharing a relatively optimistic, happy story about invasive species where it feels like we have something positive happening or that could happen, because we don't always get that. So thanks again to all of our presenters. We have plenty of time for questions. Go ahead and put those in the question and answer box in Zoom. What we can do is go through and answer just some of them live right now. And then if you all have any questions that we don't get to or they come up later, you can always reach out either to myself or Tegan and we can make sure presenters get those questions. So you can get all your green crab questions answered. From anonymous attendee, curious if European green crab was a prey item, and is there evidence that large green crabs are eating small green crabs? I think, Mary, that one sounds like it's for you.
Yeah, that's a really great question, especially because cannibalism has such a big impact on crab dynamics in general, right? So the method that we used cannot differentiate between green crab DNA that's coming from the stomach of the predator individual and green crab DNA that might be from a prey item that was consumed. But there are other genetic methods that can get at this question, and they've used them really successfully with lionfish in the Caribbean to differentiate between lionfish that are eating things and lionfish that are getting eaten. So the short answer is that we couldn't speak to this in the paper. It is kind of a blind spot for our work and for applying these methods to species like crab that have a lot of cannibalism between different life stages. But there are other genetic-based methods that can be used to kind of supplement metabarcoding in order to answer that question.
And it looks like there's another one for you right after that. Can you please expand on your methodology? How exactly did you get the stomach contents out? What kind of pipette did you use? All the details of getting the food out of the crabs.
Yeah, I should have had a GoPro on my head for this so that we could get a really close-up video of the inside of a green crab. So I actually have put all of the lab protocols into a repository on GitHub, along with all of the R code that was used to run the analysis in the paper and the metabarcoding data that we produced. And so I can drop a link to that in the chat. It's also associated with the open access paper online. If you go into the doc folder, you'll see the lab protocol describing how to do those dissections and then also describing all of the steps for the genetic lab work as well. The short answer is that we cut off the top of the carapace so we could actually see the stomach as we were working with it, and then used a 1,000 microliter pipette to do a kind of gastric lavage for the crab with ethanol.
Sounds like a lot of fun. So next question is for you, Rikke. What is the Dungeness crab population at Elkhorn Slough? Have the otters eaten all the Dungeness, and are they now preying on green crabs?
Good question. The Dungeness crab population at Elkhorn Slough is nearly non-existent. There are a few species of cankrid crabs that are at the slough, but probably not in high enough abundances to make an impact. And along the lines of the previous questions, someone said, do large green crab eat smaller green crabs? I briefly did some experiments during grad school that never really came out to be very publishable, other than what I found was large crab eat small crab, period. It didn't really matter what species they were. So I'd say any larger crab can eat the smaller crab. So I guess if the larger king-grade crabs were to have an effect on the green crab populations, they should be eating those green crabs when the green crabs are small and juvenile and soft and delicious. Although smaller prey items also take more work to catch and whatnot.
Awesome, thank you. Still have plenty of time, so leave it open for another minute or two as your questions are still formulating, because I know it always takes me a long time to think of my questions. But thankfully, Kat saved us. So, Kat de Rivera. But the number of camp crabs in Elkhorn were already low before the otters really came in a high number. And elsewhere, it's been found that more otters means more Dungeness, even though they eat them. So yeah, great point, Kat. Thank you. Anything to add as our Elkhorn Slough expert?
No, that's great. Thank you, Kat.
Yeah. And then we have another, more of a comment, not a question, but we'll allow it. Apparently Taylor Swift loves otters as well. So if anyone has a connection to Taylor, we'd welcome that. I'm sure this is the Taylor Swift green crab otter collaboration we all need in our lives. So yes, thank you for letting us know that. Another question from Mary. Mary, was it a surprise to only have had one green crab eat one of the clams?
Yeah, so we've had only one green crab consume a manila clam, which is the species that we were particularly interested in because the study was really motivated and kind of pushed forward as well by local shellfish growers, and also made possible by shellfish growers giving access to their clam beds and participating in some of the collection activities. It was a big surprise. There was a bunch of work that I did to make sure that this was true data that we were looking at. So as I mentioned, we did a lot of preliminary work in the lab where we actually took tissue from manila clam and used our PCR process in order to amplify the DNA and make sure that was working. We also mixed manila clam DNA with green crab DNA to make sure that we would still see manila clam even when it was mixed up with green crab. And then I sequenced that particular crab multiple times just to make on different sequencing runs to make sure that that was a true detection. So if that, hopefully that kind of extra work tells you how surprised we were by the observation. But again, I think that because it kind of goes back to the hypotheses that we had starting the study that we used to direct our study methods, right? So we assumed that these larger males during the summer were going to be consuming manila clam. But I think after this study, we kind of revisit those hypotheses and think that maybe earlier life stages of manila clam could be being preyed on. So not necessarily large male European green crab preying on like fully grown manila clam, but earlier life stages that are being consumed by smaller crab could still be happening, and it wouldn't have been captured in our study. And there's also been a lot of green crab observed on clam beds in the winter time. So again, our months were restricted to May through September. So it's possible that wintertime predation might be happening as well. So I think kind of the next steps for this would be really to either expand the methodology that we were using to different crab sizes and life stages in months, or to use a more targeted genetic-based method. Instead of capturing a wider diversity of prey, look only for manila clam, and there are ways of doing that quickly and more cost efficiently across a wider number of samples. So yeah, that was kind of the big reveal of the study and very much a surprise. Yes.
Questions keep trickling in, so thank you everyone for the good discussion and the questions. So another question in the same line of tidal restriction is Carcinus influenced by wave action? Although not new, the theory behind the invasion in South Africa is fairly novel. We are finding it slowly creep up the coast, so any information along those lines would be helpful. And I'll let whoever knows the most about this tackle this question, because it's definitely not me.
Not that I have a good answer, but yes, I do remember this peculiar pattern of the green crabs. I mean, they're along the California coast. You only find them in bays and estuaries. I trapped on the open coast to see if you'd ever catch any. And we know that the larvae do travel along the coastline. So why aren't they on the coast either? So as was mentioned, it's in South Africa. They have a better grip, apparently. But I don't think anyone has looked into that on the US West Coast. But maybe someone else knows of that. I guess on the East Coast, the wave action just seems like a lot less dramatic along the Atlantic there. But if anyone has better information, please fill in.
Sounds like you nailed it, Rikke. So, okay, another question from Brian Turner. Mary, was there evidence of other bivalve species? Cannot recall if any were included in your study. I know green crab are keen to consume smaller bivalves in California. So,
Yeah, that's a great question. I'm pulling up the table S4 from our study right now to see exactly which ones those were. Just because it's been a minute since I've looked at it. We observed softshell clam. We observed the one instance of manila clam being consumed. I should mention as well that when we were doing that kind of pre-study preliminary work to test out which species were going to be detected by the specific PCR process that we were using, we had an instance of a mussel not showing up, but I did not replicate those results. And so I can't say for sure that we missed them because of the PCR process that we were using, but I'd be kind of hesitant to say that they didn't have mussels in their stomachs just because of that one preliminary result we got where I put some mussel DNA in with some green crab DNA and it didn't come out the other end of the sequencing. Okay, bivalves, softshell clam. Yes, a small clam of the genus Curtiella. I'm not entirely sure what the common name of that is. And then the manila clam. So those were the three that we identified down to the species level.
so looks like we have one other kind of big picture funding question that might be a good one for everyone to say something about, maybe good question to wrap up on. This is from Rosalie. What do you think are some of the obstacles in finding funding for green crab research? In your opinion, is there something missing from funders' understanding of the problem? Or maybe anything funding agencies or granting programs can do to help make it or help move some of this work forward? Or if you don't have great ideas on that, also take any future research that you're excited about, since we know that you all are presenting on things that have already been completed. If there's anything like within your scope of work that you're excited about that other folks are working on that we should be looking out for in the future.
At the risk of sounding self-centered here, I'd say that so I left the presentation off saying, yes, there are fewer green crabs at El Con Slough than anywhere else. And then this year, during one of the annual surveys, green crabs were actually more abundant at the slough than we have seen in a long time. However, what I would like to know is, okay, so if the population is increasing at the slough and we have the otters, are the populations increasing even more in the other places? Or if someone could study the otter diet at Elkhorn Slough, are we going to start seeing a higher proportion of green crab in the otter diet as the green crabs get bigger and get to be more valuable prey items for the otters? So, but Mary, you could come study that.
I think I will speak to maybe like exciting things coming down the pipe and DNA metabarcoding and diet analysis, not particularly my work, but just in general. Moving from looking at presence absence to kind of more quantitative metrics of diet from DNA-based methods has been a very like complex thing that people have been working on. And there's been a few papers out that kind of speak to that. And we did a little bit of quantitative modeling in our study for this, but I just want to highlight that even though my results were really focused, the results I presented here were focused on presence absence of prey. I know it can be extremely helpful to also be able to quantify prey that's being consumed. And there are kind of post-analysis quantitative models that you can do to show that. And that part of this field is pretty exciting and it's moving forward pretty quickly. So if you were looking at the results and thinking, okay, well, when I'm making decisions, it's more important or it's more helpful to know more quantitatively how much is being consumed. That's kind of the cutting edge of this particular subtopic. And it's, yeah, really just improving month by month and day by day. And there are methods of doing that are being worked on right now.
I can also highlight some green crab research that's happening. Another chapter of my PhD is in review right now. With green crab, we live in the world of catch per unit effort, which is a great measure of species density, but it can be quite limited. Particularly, it's challenging to kind of like disentangle natural mortality from harvest mortality. So I've made this basically like big Bayesian model that's a big kind of data integration exercise to estimate kind of like absolute abundance and also absolute kind of size selective harvest rates. So yeah, happy to, if you're interested in that, happy to share when it gets published.
Well, great. Thank you again to all our presenters for the great presentations today. Thank you everyone for attending. Tegan, I'll hand it over to you for any NAISMA wrap up. And again, thank you, NAISMA, for helping organize and host. We really appreciate it.
Thank you, Tim, and thank you to all of our speakers. I really appreciated hearing your presentations and highlighting this important work, and wish you all the best with your future research efforts. I just have a couple of final slides to share today. All right. And so we do have a great lineup of free monthly webinars continuing throughout the rest of this year. So next month, we have a panel discussing efforts to combat invasive species to protect unique island ecosystems. So that one's going to be really exciting. And then in October, we're having our webinar a week earlier than usual. So on October 8th, we're going to have Dr. Lynch from UC Davis talking about lessons learned from managing the polyphagous shot hole borer in California, and how those are helping management efforts in Western Australia, which is where I am originally from. So I'm excited to hear about that talk. And then lastly, we are partnering again with RISC to host the Invasive Species and Climate Change Conference on December 9th and 10th. This is a free virtual conference, and this year's theme is Better Together, Empowering People and Partnerships. This event was a huge success last year, and we're very excited to be hosting it again. And you can register by scanning this QR code or by visiting our website. And again, just want to say thank you to all of you for joining us today. We look forward to seeing you at an upcoming event soon. And in case any of you missed part of this webinar or want to share it, we will be posting it to NAISMA's member area, as well as to our YouTube channel once it has been processed. So thank you all very much, and I hope you enjoyed the rest of your day.